make this amazing. This is going to be an amazing session, everyone. Okay. Yeah. Are you ready? Session 2. All right. Okay. So my name's Janet Reid. I'm from the CSIRO Biofoundry in Brisbane in Australia. So I'm animating session number 2, which is on the DNA build, automating DNA build. I'm hoping that everyone online can hear us as well. So we have 2 presenters online that will be last up, but we have 3 presenters here with us today. So thank you very much to the presenters. They span a range of perspectives, and the group includes a presentation that demonstrates the utility of biofoundry automation for purposes outside of synthetic biology pipelines. So I'm really looking forward to this session. So first up, we've got Pablo Carbonell from the, he's a senior reader in the computational biology at Universitat Politécnica de Valencia in Spain. Correct. Pablo is currently a visiting scholar at I2 CISBio as well. He's got an interest in automated design for metabolic engineering in SynBio. And he's contributed to the development of a number of computational tools. And today he is going to present on a couple of those. So one of them is a really great example of collaboration across biofoundries. So really pleased to hear about that work today. And that actually spawned out of the GBA, I believe, that particular body of work. So thank you, Pablo. Please take it away.
Thank you. Thank you for this nice presentation. I'm happy to be in this very interesting meeting of the GBA. So yeah, I'm going to talk about some software platforms that or initiatives that we have been developing and trying to make it the transition between what has been the first session about software and the second one that is more about the automating the build stage. In Manchester, at the SynBiochem Biofoundry, we came up with, it was in 2018 where that was published, this workflow that you see here, which was an effort in order to identify all the different steps that you can have in a typical DBTL cycle, and with the purpose of trying to automate every step. Obviously, you can go deeper into the details of every step, and you will find more and more stages inside them. But I think that this covers what typically is performed during one iteration of the DBTL cycle. So, in order to perform this automation, the first step was normally to try to have everything that is computer-based, software-based, well connected, interrelated in a way that you can, both the design and the learn stages, make to talk each one with the other, and then have the tools that like pathway design or enzyme design, DNA part design. So these were all individual tools, but they can exchange data. So that took several years, but in some way that was the easy part of automating the DBTL cycle, because we were just working with software. Then the point is this something that really works? In our case, our test bench was for producing flavonoids. What we did here was to design initially a library using all of these software tools that I just showed you, and then we performed the build and test and learn. And based on that initial iteration, we went through a second iteration here, which finally we found that the titers increased substantially from our initial run. And that was like a proof of concept. We saw that we can compress a lot the design space, just to focus on those factors and those combinations that are really relevant. So But now the question is, if we want to automate the build stage, what are the challenges? First of all, we need to make sure that we can make the integration of these two steps, but we found usually a lack of interoperability of tools. There are lack of standard formats for data exchange. There are no universally agreed software solutions for an automated lab or commercial solutions that often do not scale well. So at the end, what you usually found is tailor in-house solutions for each biofoundry, and frequently will reinvention. That was something that came up usually in the discussions of the GBA software group. We look at the landscape of the hundreds of different tools that different biofoundries, different labs have been developing. Here you have an overview, but obviously there are many, and all time many more are appearing. So then we said, okay, let's try to do our first steps into trying to address this challenge. And that was when the idea of developing the SynBio Python package started, which actually it was and still is a really nice collaborative project between different members of the GBA, and also is open to other people from the community. And basically, the SynBio Python initiative is inspired by BioPython, which is a project that has been developed by the bioinformatics community with the same goals, that is increasing the reliability due to community testing, increasing the reusability and interoperability, and the community adoption, which is something that we were discussing in the previous session in the workshops, that are some of the problems that we have nowadays. But in the case of the SynBio community, there are some specific requirements besides the one that usually are found in the bioinformatics community. We need tools for DNA design, and we need software for automation and robotic equipment. So that's the idea of the SynBio Python. And our initial focus has been on format conversion, on CAD tools, and also providing automation library. As in the previous talk, you saw already some examples about it. So this is still an ongoing project, and we hope that you will hear sooner than later about SynBio Python 2.0. Then in parallel, there's another initiative that has been developed in the last years, which is the Galaxy SynBioCAD project, which is also in the same spirit, but here the main focus is about having a platform that allows you to interconnect these different tools that might be based on SynBio Python, might be based on some other in-house software tools that you have. How we can interconnect them together and create a full workflow, what we call an end-to-end metabolic pathway design and engineering process. So that all of these steps, like selection of strains and targets, design of DNA parts, generation of work lists for liquid handling robots, are all addressed, so that you end up having the instructions that should be uploaded into your OpenTrons, for instance, and everything by using standard formats like SBML and SBOL. So you can see here the current workflow that is being used. You can see that many of these tools are the ones that I mentioned before that we were using in Manchester, plus others that have been also developed. But here they are interconnected between each other through different standard formats like SBML or SBOL or Scilab, which is for the part of the lab automation. So you can start just by providing your initial targets, the molecules or the family of molecules that you want to produce, and then let the system to generate all of these, select the enzyme, the pathways, and generate the DNA parts combinations. And finally, run it in the robot. So as I said, this is just based on workflows, and actually you can go into the detail of them if you want to set the different parameters. So there's this freedom if you want to actually, for instance, perform the retrosynthesis for the pathway search in more or less depth. Or, for instance, another main step, which is the pathway analysis, which is the one that tries to sell from what are the initial candidate pathways, tries to find out what will be the most promising pathways. And for that purpose, there are different types of features that you can score, like, for instance, energy or fluxes and so on. So everything is put together in order to try to find what will be the optimal design. And then we have the actual implementation of the design in the OpenTrons. For this first implementation of the Galaxy in BioCAD, we have been working with the basic assembly in order to use the DNA bot protocol, which was developed by Imperial. And the idea was to validate the workflow in two different labs, one in France and the other one in Imperial, and then see what are, the reproducibility of the results, and also what might be the differences because of the specific particularities of each different lab. So it's more a way of assessing what can be done and how successful this can be. So this is still open to more and more modules that we can add to it. And then what is probably the main challenge, if we want really to automate the build process, is to make these workflows to work on a, let's say, biomanufacturing fashion, so that we can run it in parallel for multiple projects, and also we have ways of performing rapid prototyping. So in that way, this is a proof of concept of that was performed in Manchester, where we selected around 25 different targets, and all of them have to be produced in the biofoundry in parallel. And you see here in these chronograms how each different target proceed through the different steps or stages of the DBTL that I show at the beginning of my talk. And obviously the challenge here is that you need to decide about resource allocation, when the robots are available, and assign slots depending on the priorities. So it's like a new layer that appears now in the automation of the biofoundry, where what we want is to actually schedule the different processes like if you were in a manufacturing process. So from my point of view, where we are going now in this direction is towards what is called the smart biomanufacturing. Actually, usually, probably you have heard about smart manufacturing, which is something that you heard from the car industry, or you heard from the electronics industry, but also we can think about it for the smart manufacturing. And basically what we need is to adopt the ideas of the Industry 4.0, automation, machine learning, Internet of Things, interconnectivity in the lab, so that the different equipments can exchange data, and system integration, so that we can work on optimal experimental design, modeling, and simulation. One of these first steps towards the smart biomanufacturing is developing digital twins, a biofoundry digital twin, which is something that now we have in all types of manufacturing industries, and we should also have it here in the biofoundry, which is a digitalized real-time virtual representation of the biomanufacturing process. Because this is going to allow us to simulate and reproduce different scenarios and try to find out where are the bottlenecks and what has to be optimized in the process in order to make it more efficient and deal with this high demand of different targets, which is something that we should expect as we are moving more and more into the bioeconomy, and there will be more and more demand of producing bio-based chemicals. So we are working now on this. We have a couple of grants, and actually we are hiring people at the moment. So we are expecting to have some prototype soon of a digital twin. But just to conclude, a quick example of how we can use this virtualization of the DBTL process. For instance, we can do this type of simulations where you simulate all the different pathways, all the different combinations that exist are currently known that can be produced in E. coli, for instance, and then try different types of designs with more or less number of experiments and see how this is going to affect the efficiency of the design. And in parallel, also, you can simulate the processes. This is why we are talking about a digital twin, and see how this is going to impact the actual titers that you are going to get in your strains. Obviously, this is just a simulation, but you perform it based on data that are already available on databases, and statistically, what we get here can give us some guidances, some design guidelines about how to perform and what could be prioritized on your different targets. So just to conclude, then we have seen that the first step was to integrate design and learn towards DBTL automation, and some recent platforms like SynBioPython, Galaxy SynBioCAD, Biofoundry Palette, PyLab Robot that are appearing are providing better interoperability with the automated build stage. And for the next step probably is to work more on the application of this idea of the smart biomanufacturing. But to do that, I think that one challenge that we have now is that we need a better understanding of the effects of the experimental and design factors when scaling up is needed. I mean, the challenge still is that everything that we are simulating then has to be also applicable when you work on the fermenters, not just what happens in the lab, which is probably the data that you have available from the DBTL cycle, but also when you're going to perform the scaling up. So something that we need to work on this, and I think is going to be the challenge for the next year. And with that, just thanking my founders, and I will be happy now to take your questions. Thank you for your attention.
I'm so sorry. In the interest of time, we won't take questions from the guests in house, but we'll take a question or two from the people who are online. But I please encourage you to approach Paulo as we meet over the next 2 days. And thank you so much for a really great presentation, some really good information that you've presented us today. So thank you, appreciate it. Thank you. Hey. Momentary pause. So next up we have Lachlan Jake Monroe. There we are. So Lachlan, thank you very much for presenting today. Lachlan's from DTU. He's a lead automation scientist. Sorry, I'll step forward. He's a lead automation scientist from Novo Nordisk Foundation Center for Biosustainability. And he's made a leap from molecular pharmacology into synthetic biology, now leading that new automation group. And during his recent postdoc, he was looking at membrane transporters in the context of drug and compound transit across cell boundaries. And to me, that sounds like a really relevant thing for the AMR problem. But today we're going to hear about generating targeted variant protein libraries with speedy genes. So thank you, Lachlan.
Thanks, Janet.
Please.
Yes, I will plug in. It's on the other side, but we'll manage. So while this loads up, it does. So it's great to be here. Very nice to be at an in-person conference again. One of the side effects of COVID is that I'm completely unaccustomed to time zone changes. So if I lose my train of thought at some point, I apologize. We'll do our best. But yeah, so I'm going to talk about a sort of platform protocol workflow that we have set up at DTU. Called Speedy Genes. It was originally developed by my old supervisor, Douglas Kell, and Neil Swainston, whose name has popped up on some of these other computational synthetic biology tools. But it's a methodology for generating protein libraries in a very targeted way. So I'll give kind of a pretty broad overview of the method. I won't go into too many nitty-gritty details. I have a poster as well later. I'm around for the next 2 days. If you're curious, or you want to ask anything specific, please just come and talk to me. I'm more than happy to talk about it. Okay, so the problem of protein engineering is that the landscape of potential proteins is sort of unfathomably large. Protein of length n, there's 20 to the power of n potential variant sequences. So for any sort of reasonably long protein, we start to get into number of atoms in the universe type numbers. So it is sort of completely unfeasible to do any kind of search of even a tiny fraction of the potential options. So you need to kind of have a bit more of a targeted approach to do any kind of protein engineering where you're trying to improve the function of a protein. So you can kind of broadly take two approaches to protein engineering. One of them is the sort of rational approach. So you have some hypothesis about how the protein's working. You maybe think there's some particular residue interacting with the substrate in a particular way. So you can generate a mutation to test that hypothesis, and then you can screen the mutant to see if you were right and potentially improve the protein in that way. This is obviously quite a low throughput method, and it requires you having sort of good knowledge about how the protein's working. And then there's this sort of classic directed evolution methodology where you don't need to know how the protein is working. Instead, you sort of just generate a bunch of diversity. So either through random mutagenesis or an error-prone PCR. I put ALE up there as well, which is adaptive laboratory evolution. ALE is sort of all three of these steps looped into one thing. But broadly, they all work by just generating random diversity in the sequence. Then you have some kind of screen or selection assay. So potentially you can have a growth selection, or you can run it in FACS if you've got some kind of biomarker. You can also just do plate-based screening and try and sort of map out a little bit based on random variation what different changes are doing to the function of the protein. And then you pick the one that works the best or had the most effect on improving the function, and you can sort of loop through that again and again iteratively to generate a more protein with improved attributes that you're interested in. So this is quite an effective method, but there are some obvious sort of shortcomings and problems. So if you're just randomly mutating things, most mutations are going to be negative on the function. So in general, you're trying to find a very specific point in the protein landscape, and most directions you go are going to be away from that, because presumably starting with an enzyme or a transporter or something that is already doing the thing you want, and then you're trying to move it up a pretty specific pathway to become more efficient at that specific thing. You also have the risk of getting stuck in like a local maxima, which is sort of a concept that is talked about a little bit in synthetic biology. But if you visualize the protein landscape as a kind of mountain range, you can think that there would be points where you get to a little peak that is not actually the top, but any direction you go from there, any direction you go from there is actually going to reduce the function of the protein. And once you're there, if you don't have a method to sort of get out of there, you can end up stuck in sort of a local maxima, as we call it. And you also have the problem that you can't find epistatic effects. So often you'll have mutations that on their own don't do anything or are negative, but in combination with other mutations, they have positive effects that can be sort of, how would you say? So this is what I warned you about. Where it's more than additive. What was the word? Cumulative or synergistic. Thank you so much. Yeah, so you can have these synergistic effects that you won't actually catch if you're just doing sort of single mutations one by one. So that is where this method that I'm going to talk about comes in, which is called speedy genes. Broadly, it is a way to synthesize genes and proteins using overlapping oligonucleotides. So we have some programs where you can design a set of overlapping oligonucleotides, and you basically just run a PCR of those. You synthesize, you can do it in one hit if it's a small enough protein, or you can do it in separate blocks depending on the length. And then you combine the blocks and run it again with the terminal end primers, and then you end up with your full gene based on the oligos there. So for just synthesizing a defined sequence, it's probably not the most effective method, but I'll get into this sort of applications and how we can expand this down the line. I will also mention another tool that we use a lot in Speedy Genes, which is called Codon Genie, and this is not great. So this is basically a web app where what it lets you do is select residues that you want to include, and then it will spit out the degenerate codon that encodes those residues. So you could, for example, say, okay, I know this position is got to be a hydrophobic residue. I know it has to be one of those. And I'm just going to say I want all the hydrophobic potential amino acids to be present at that residue. Same thing for polar or different combinations. Okay, so this sort of base speedy gene synthesis from oligos concept, it can be used to kind of do different effects or different approaches to generating variant libraries. So one option is you could make just a very large diverse protein variant library where you go along a sequence and you pick specific residues and introduce degenerate codons to get tons and tons of variability all in a single pool. So that would be, just going along and introducing these degenerate codons, either all 20 amino acids or subsets of that, and synthesizing a big pool of protein variants. The other option is down here underneath that. You could do targeted sites of variation, where if you mix the oligos in different ways, so you could have the wild type, and then you could mix, do I have a mouse? Nope. You could mix the wild type plus this single oligo here, so you're going to get just all the variants at that position. You could do the same thing with this one, and so on and so forth. So you get sort of one by one variants at set positions. And then, of course, you can start combining those as well. So you can do sort of systematic one by one introduction of variation. And then on the right, you can also do fully targeted libraries. So you could make, if you say have 5 separate amino acids and you want to keep them separate at specific positions, you can mix the oligos so that you get all the different 5 variants at that position, all the 5 at the other. And then, of course, you can run all the combinatorials of that as well. So it's quite a powerful method where you can do sort of lots of different options for making these libraries, whether you want to have them all pooled together, you want to mix them in, you want to have them separate to screen and actually target in on the effects if you're going to do machine learning down the line. Alrighty. So the workflow that we use. So there's a web app called Mutation Maker, where you basically just plug in your either DNA sequence or amino acid sequence, and that will spit out the oligos and the variant oligos, depending on what you tell it you want to make. So then you'll have your list of oligos, and then there's some Python scripts that I've written that will generate the workflows for the robots. So you just plug in the Excel sheet that comes from the Mutation Maker into this Python executable, and that will spit out the workflows for the different robots. The workflows are, so we have an echo, and that will do the combinatorial mixing of the oligos and load the master mix into the PCR plates. And then we run everything downstream of that. We have an Opentron, so it'll run the PCR, do the mag deck cleanup, mix for the second combination of PCR. And then we're working on transformation because they have a heater shaker. So that will be something we're hoping to also integrate there. So that's kind of the broad strokes. I'll give a sort of example application that is something we've done recently using this system. So we do sort of just synthetic biology cell factory stuff. And one of the projects we have going at the moment is an E. coli strain producing, overproducing tyrosine. And we wanted to try and improve the yield from this. And so quite an effective way to improve the yield of a cell factory, and I'm sure this is not news to anyone, but that is to actually increase the amount of product that's getting exported from the cells. You can do that by just overexpressing an export transporter, but that tends to have some negative effects. The cells don't really like pumping too many things into the membrane. So another approach is to have the same amount of protein, but in some way optimize it to either speed up and export more of your product per transporter, or to more specifically export the product instead of a wider range of things. So in E. coli, YDDG is an amino acid exporter. And so the project was to optimize this for tyrosine export and see if we could both speed up tyrosine export and do tyrosine over the other aromatic amino acids, because it'll do phenylalanine and tryptophan as well. And so we also have a nice alpha fold structure now, so we can see what it looks like. Okay, so like I said, for the rational approach, you kind of need some sort of like specific knowledge. For sort of classic directed evolution, you need no knowledge at all. But this was a situation where I think a lot of things are now, especially with the big data sets coming out and AlphaFold. We actually had a bit of knowledge and we had some data, but we didn't really have any actual understanding of what that was telling us. So what we had was a pangenomic data set, which is basically people go out and you get E. coli from hospitals and from wastewater. And it was basically pooling all the different sequences of E. coli together. So we can pull out the YDDG sequences from there, and we can just go along and see where are things showing up in nature, where are we seeing amino acids that can be varied. So this picture here, I don't know if anyone's seen this before. There was during World War II, they were looking at where they should reinforce the aeroplanes. And so when the aeroplanes were coming back, they were looking at where there were bullet holes, and they're saying we should reinforce it where there are bullet holes. And then someone came along and said, no, we should reinforce where there are no bullet holes, because the ones that got shot where there aren't any bullet holes, they went down. The ones that came back where the bullet holes are, they're fine. They can have a bullet there. So kind of maybe a little bit of a stretch of analogy, but we're looking for things where we can send the protein out and it comes back with a mutation. We think, it can tolerate a mutation there. It still functions if we do mutations there. So we looked through this data set, and 60% of the residues showed some kind of substitution. And so of those, we managed to get 45 positions into the design. And I did exactly as I talked about with the codon genie. So if I saw a residue, it could be an alanine in the wild type, and then cysteine or tryptophan showed up in the data set. Those were the 3 potential amino acids that went there. So it was really combining mutations that existed in nature with the wild type and pooling them all together to see what we got out. And this is just sort of an example of how it would look with the sort of degenerate codons. So yeah, I ran this through the workflow. And then I had this pool of library. I think I mentioned it's 10 to the 23 was the final size. I'll admit this was partly me just wanting to see a big number on the screen, because obviously we're not going to get that many transformants, but that was the library size in principle. So yeah, this was transformed into a E. coli, where we'd knocked out the YDDG, so it was lacking the native transporter. So this was going to be the main exporter. And then we did a selection growth in a toxic analog of tyrosine called tyrosol. I'd like to show you guys sequencing data. We may patent something, so I'm not allowed to, but I can give you kind of the broad strokes of what we found. But basically sort of specific things when we did this selection assay became enriched. And again, I've got the nice alpha fold structure here, and the red regions are the regions where we saw big divergence away from wild type. So this tends to be in the sort of extracellular loops, which to my mind suggests we haven't actually affected the substrate specificity, but what we've done instead is probably increase the Vmax by kind of increasing the flexibility or the floppiness, I guess you could say, of the protein. So we have these sequences, and the other nice thing about speedy genes is once you have your oligos, you have them. So we can actually iterate quite easily. So if we know these are the regions that we're interested in now at this point, based on this first selection, we can actually lock down everything else by using the wild type sequence for everything that wasn't showing up as enriched, and then really focus on just using the degenerate codons at these regions. And we can even go back and do more specific subsets of these degenerate codons again. So yeah, that's basically all I had. I've just kind of dumped all the references that are relevant up here. So there's Speedy Genes, the original paper came out in 2014, and then Speedy Genes XL was just published this year, which is, both of these has very nice kind of step-by-steps and explain the method in quite a lot of detail. A group from Merck did Mutation Maker, which is a software that I'm using that basically does the same thing. The kind of second thing they say in the title of the paper is open source, but they had it up for about 3 weeks and then they took it off their GitHub server, which was quite irritating, but luckily I've cloned it. So please go and get it from my GitHub, because I think that's a little bit out of order that they did that. And then also Codon Genie, you can read more about the software and how that's actually working if you're interested. So yeah, that's all I have. Thank you so much. As I say, please come talk to me about this.
Thank you so much. That was a great presentation. Thanks very much for that, Lachlan. And our next presenter is Courtney, who I believe is in the room. Ah, there we are. Let me just bring up your slide. Yeah, and so thank you. Today we've got Courtney from DAMP Lab. So I'm very pleased to present another Biofoundry manager. So thank you. I'm a Biofoundry manager. Yeah. To the podium. So from DAMP Lab in Boston University, Courtney manages the clinical testing lab, and today is going to discuss the rapid and successful pivot that they've made into integrating clinical testing into the synthetic biology platform. So welcome. Thank you. Thank you.
Okay, so as Janet just said, my name is Courtney Trethaway. I came to the DAMP Lab from the clinical testing lab at Boston University. So I was managing that before the transition over into the DAMP Lab. So I'm here to talk about high throughput clinical infrastructure applied to DNA assembly automation. So just a little bit of background for anyone who may not know on the DAMP Lab. The DAMP Lab was founded in 2017 by BU professor Doug Densmore, who is, I believe, in this room somewhere. And the history behind the name is he was thinking about his vision for the lab, and he was saying, it's not really a wet lab, it's not a dry lab, it's a damp lab. And then after that term, he came up with the, in the top right corner, you'll see the acronym. So design, automation, manufacturing, and processes. So 2 years into the DAMP Lab being at BU, it became a core facility at BU, which established itself long-term to offer services to the BU community. And then in 2019, the GBA was there was the inception of the GBA, and we joined that. And then also early 2020, joined the International Gene Consortia, which some people know 2020 as some other major world event, which was COVID-19 hit. And that did obviously affect everyone. And so BU decided that they needed to be able to offer high scale testing to the entire BU community. And so this effort was put into place that they would build this clinical testing lab to offer those services. So it tested all 45,000 people on the BU campus, and the lab itself was developed in under 3 months. So a very impressive feat that they achieved there. And then for 2 years, it operated independently. And then in 2022, the university decided to ramp down its asymptomatic surveillance COVID testing to only do symptomatic testing. And at that point, they decided to put the clinical testing lab back under the DAMP lab. And so right now we're in the process of transitioning all of the clinical testing lab infrastructure into the DAMP lab. And so the DAMP lab is an academic foundry at Boston University. It's located in the newly built Rajan Kilishan Center for Life Sciences and Engineering, which is a unique building at BU because it is one of the only buildings that is multiple departments. Most of the buildings are just one department. So it's either engineering, it's biology. This is anything and everything. There's neuroscience, there's synthetic biology, microbiology. And the Biological Design Center actually lives in this building on the 4th, 5th, and 6th floors, which the DAMP lab is also a part of. So it's really nice to be in this melting pot of different biological ideas with different synthetic biology and microbiology labs. And the vision for the DAMP lab is a fiscally self-sustaining automated core facility for high throughput, high quality, and cost-effective synthetic biology. And then the mission is to create robust and reproducible automation workflows on an open source platform that allow for faster, more scalable, and more accessible research that can be utilized by not only academia, but also industry. And so this video is the CTL workflow. So over here, this is where sample intake would take place. So people would have to scan in all the samples and activate them with heat because they are potentially infectious. So as they come in, they're inactivated. They're put on our first liquid handling robot where they are aliquoted into a 96 deep well plate. And then from there, they go into RNA extraction. We have dedicated robots for RNA extraction as well. So 4 96 deep well plates would go onto the extraction robot. The method would run for about an hour and a half to 2 hours, depending on how many plates you would put into the workflow. And then 4 96 well plates would come out. Those 4 96 well elution plates would come out, go onto other Hamilton robots that we had for qPCR prep. And so 4 96 well plates would be condensed into 1 384 well plate. And the reason that we did this was to reduce the need for the qPCR machines, which we use Quant Studios. So the less plates that you have going through the system, the less Quant Studios you need. Because if we left them just in 96 well plates, we would have needed probably at least 12 to 15 of the Quant Studios where we only needed 5. And we had 7 liquid handling robots in the clinical testing lab. So this over here is just a workflow of how the lab was designed. So samples would come in, they would be accessioned and decapped. So this would be where all the inactivation would happen. And then this is the sample aliquoting robot, which is right here. We had 1 sample aliquoting robot, and then they would move to RNA extraction where there were 3 Hamilton robots that would perform the RNA extraction. And then we had 3 additional qPCR Hamilton liquid handlers that would perform the qPCR prep, and then the 5 Quant Studios that would perform RT-qPCR, and then we would do data analysis with that. And so this is just a little snapshot of the impact that the clinical testing lab had in Boston and the surrounding area. So there are many Boston University papers that published about the clinical testing lab, and NBC came and did a story, and also the presidential administration in 2020 did visit the clinical testing lab as part of their effort to do more investigation into how best to implement systems throughout the government as well. Because there had been a few schools that had built these labs and they wanted to go and see a small subset of them, and we were one of them. And so since it was one of the first ones around. That was why the presidential administration came. It was definitely one of the first in Boston. And so a few other Boston schools, including Northeastern and Harvard, created their own testing labs afterwards. And so this is some data that I compiled from the clinical testing lab. So average cumulative sample turnaround time was around 15.29 hours. Anyone that has gotten a COVID test anywhere else knows that's not bad. It's taken me days sometimes to get COVID tests back. So it was very good, and it enabled the university to stay on top of any outbreaks or any potential breakouts that were happening on campus. Average cumulative positivity rate was 0.65%, which isn't really too high compared to the average for globally. And then you can see, so this graph up here, like I was saying, we stopped doing asymptomatic surveillance testing. And so this is since October 2021. So from October 2021 to around here, this is all the surveillance asymptomatic testing. So the highest peak in positivity that we had was around 14%, which these are all, in theory, healthy people. And so we would expect them to not have COVID-19. This, however, is since we started doing symptomatic testing. So that is why this positivity rate has spiked so high. And then it kind of also correlates with this graph here. So this is samples run per day. You see these little dips. This is just for winter break and summers when people aren't as on campus. But this first year, it was around, we were doing around 6,000 to 7,000 a day sample COVID-19 tests, which was possible because of all of the automation equipment that we had. But then in 2021, with variants and other such factors, they decided that we needed to be able to test even more. So we started averaging around 9,000 a day. And the reason we were able to do this is because we started pooling our samples at the beginning of the process. So rather than putting one sample in, as we had done the whole time, we put 5 into one, push them through the whole process that way, and we're able to deliver about 5 times the results. So it definitely was a really great way to be able to scale up our protocol. And then this is a little snapshot of how many we've done since doing symptomatic only, and we peaked around 200. So this is kind of a perfect graph to represent that we have a lot of bandwidth in this automation workflow that we are going to implement doing other things. Compared to the 9,000 we were doing before, we're down to 200. But total samples run is 2.3 million samples and counting. So it's still pretty good. And then, so some benefits that we learned from automation infrastructure. So precision is obviously key, especially in a clinical lab, but also in any sort of synthetic biology or microbiology lab. This is a little chart from the Hamilton website, which we use primarily Hamilton liquid handlers. So this would be pretty accurate for our liquid handling robots. And then doing high throughput testing proves the importance of reproducibility. We were able to do so many tests because we had one assay, we had one workflow, and you're able to do it over and over again. And that's why we were able to keep the BU community healthy and safe for all of COVID. And then also the ability to scale up and down, like I was mentioning earlier with pooling, it's important to be able to do that. And I think that is something that will adapt to future workflows in the synthetic biology realm, because it's important to be able to adapt to what someone's giving you rather than what you started with. And then resource utilization, it's important to be able to decide what you want where, basically. So that's why we implemented the dedicated robots for RNA extraction and qPCR. So we could have hypothetically put those sample aliquot, RNA extraction, and qPCR all onto one robot, but the throughput would not have been as good. So it was a way to utilize our resources, and also you're able to use your time somewhere else. So someone who develops a workflow and would send it to the DAMP lab doesn't necessarily have to spend all of the time doing it because they send it out to get tested. Oh, okay, automation infrastructure limitations. So throughput versus modularity, this is what I was saying earlier, like it can be designed to do a lot of one thing or many things at once. So that's why we had the dedicated robots. There is a high upfront cost. So designing the lab in 3 months was it was a very hefty feat, and it definitely wasn't financially cheap either. But I do think that it was worth it in the long run. And now we have all these resources that we can apply to other things. R&D costs and just time to develop assays like this aren't cheap. It's not easy. You have to, we're a CLIA approved lab, so that process in itself takes a very long time to develop. And then cost benefit analysis. We were talking about this in the workshop this morning. Does it make sense to automate something? You have to kind of take into consideration not to automate just to automate it. You have to make sure it's a need. In this case, in the clinical testing case, it was absolutely a need, but in future workflows, it may be a need or more of a desire to do so you don't have to do it manually. But I think it's important to consider your costs too. So for example, in a DNA assembly workflow at the PCR step, if you're going to automate it, you have to add in additional dead volume for your master mix. You may not necessarily have to do that if you run the process manually. So you have to take into consideration, how many samples am I going to be putting into this? How many people are even going to use the service? Does it make sense to do it? Oh, and this is just a little, it's the Hamilton. And so our next steps are, we are currently automating the DNA build process in pieces with manual intervention. So this goes along the same thing of the cost benefit analysis. Does it make sense to automate it? Maybe it's easier just to have a person take the plate and move it somewhere else. At this stage where we're at right now, we're evaluating pilot project proposals at Boston University. So essentially we put out a call to all of the university and said, what do you not want to have to do manually? What would you love to just send out and not have to think about? And a few of the things that have been the top winners that so far are NGS library prep, DNA assembly, and mini preps. Those are the things that people really think there's high value in automating at this high throughput scale. And then also maintaining the ability to operate as a clinical testing lab and expand clinical services offered. So we're currently working on validating monkeypox, a monkeypox assay and a influenza B assay to provide to not just the BU community, but also to externally as well, which has a high value in the scale that we could test at and also for the university itself. And then a little more distant future is to increase hands-off automation of the DNA assembly process by adding additional modules to robots and linking together smaller protocols to reduce manual intervention. So this would basically be, we've decided that it's worth it to automate those steps. So rather than having the person move the things around or take a plate off to seal it, we've decided we need to add in a module to seal the plate. We decided we needed to add in a non-dick thermocycler. And then executing the pilot project. So this goes along with those ones earlier, the DNA assembly, the NGS library prep. So deciding exactly what people need and making sure that it gives off exactly what we wanted it to. And then incorporate software tools to streamline services, scheduling equipment, data delivery, et cetera. This is definitely important, especially scheduling equipment and service requests. I think that also on a software standpoint, the clinical testing lab is using eLabs, which is not necessarily open source, and the DAMP lab would like to be more of an open source network, but it's harder to be open source as a clinical lab. So that's another hurdle that we are facing. And then also 2023, we're holding a workshop, the IWDBMA. So in June of 2023. So if you want to come, let me know, hit me up later. We have a poster that we're doing later, so you can come find our poster. And then this is a quote that I pulled from the developers of the COVID lab. They wrote this paper and I thought it was really applicable. So The Microlab Star can be modified to execute multiple tasks due to the flexible deck layout and modular components that lends to developing unique and integrated automation protocols. Additionally, the instruments can be adapted to various assay and sample preparation protocols when large-scale COVID-19 testing is no longer needed. And so that's where we're at. We're at the point that large-scale COVID-19 testing is no longer needed. So we have this large amount of infrastructure in place to do this high throughput testing. And so we're evaluating currently how best to use it and working on automating things further on the liquid handling robots, the Hamiltons specifically. A lot of our processes had been automated on Opentron before, but now we're trying to scale up and do them on Hamiltons as well. And these are my acknowledgements. And I just want to thank all of you also for being here today.
Thank you so much, Courtney. That was a great presentation and a really good use of the automation platform for multiple purposes. So next we are going online, I believe. Let me close this one. I'm going to let the wonderful tech team guide me a little. We've got Peter. Hi, Peter.
Hello, can you hear me?
Perfectly. Let me give you just a tiny introduction. So Peter is based at the Edinburgh Gene Foundry. He's a computational biologist and develops and maintains open access collection of web applications and open source software libraries. So he's going to be talking about the EGF platform today. EGF is one of the leaders in the sort of automated platform setups. If anyone has ever seen the EGF setup, it's pretty impressive. So thank you very much, Peter, for presenting today on their behalf. Please take it away.
Thank you very much. I'm going to do a screen share. Can you see my screen?
Yes, we can.
Excellent. Okay. So yes, I was introduced. Thank you very much. My name is Peter Beek, and in this presentation, I'm going to talk about the setup of our foundry, because there are many new GBA members, and I think there are a lot of information that they can use as well. Then I'm going to talk about our workflow and our software, and I will invite everybody to try and use our free and open source software for collaboration or just for your own foundries. So here you can see a photo of the foundry and also a layout of the foundry. So we have our own software that we use to assist around the design and the operation of the platform. We use the GBEI ICE database to store the samples. And we have the Thermo Fisher Momentum software that orchestrates everything around the platform, what's happening. We have 3 industrial robotic arms that move around the plate that we use and other material between the various equipment that we have on the automated platform. And like all other foundries, our goal is to provide a fully automated platform that can scale up and automate processes and build processes for the researchers. So as I said, I'm going to explain our workflow and then going into the software side. So there are 3 main steps in our main service, which is DNA assembly design from part. One is the DNA design and the planning of the procedure, the actual assembly in the lab, and then quality control and verification of the product that we get. So the DNA design phase, the customers usually come to us either with a combinatorial assembly plan or with a desired sequence that they wish to get. So we're evaluating these sequences with our software DNA Giselle that can both flag problematic patterns such as monomer repeats and also modify them in an automated manner. We screen both the customers and the sequences, the project, and the construct themselves for biosafety. And then at the next stage, in the assembly planning stage, we actually draw up the assembly plan, which is essentially an Excel sheet or some kind of table telling what constructs are building from which part, And we have various tools like DNA Weaver and many other tools that can assist for different kinds of assemblies. And as a final step of the DNA design, we do a cloning simulation, which basically simulates the GenBank final plasmid files for each of the constructs that we are making. The final check and checkpoint for the customer. And once we get the green light for this cloning or the procedure, then we prepare the actual files for the robot and the instruction files and the nice report on the assembly procedure using our own software, Plato. And at this stage, the work is handed over to the research technologists from the wet lab team, who are going to use these files to start the procedure, which is mixing the part and the enzymes on the right with the lab side acoustic transfer machine, incubate them, and then transform the product, the construct, the plasmid construct into bacteria using the take-on freedom liquid automated liquid handler, and then transform the cells, incubate them, and then pick the colonies with the QPix colony picker. Once you got these colonies, then we purify them using, again, the take-on freedom Evo machine, and we go on to the verification stage, which is currently with fragment analysis. We have, again, our own software that can take the data, zip file basically, from the fragment analyzer, take the reference files, and telling you what samples are good, what clones are good, and what are not. More recently, we have acquired a MinION sequencer and set up in the lab to work in an automated, semi-automated manner, and develop the corresponding pipeline that we are going to publish very soon that can just, in a one-step way, analyze the data and provide a nice report on each of the barcode sequence plasmid that we have made for an extra cost. So again, this was the linear timeline of the workflow, and this is the actual high throughput platform. See, you can see where each of the individual elements are on the platform, the dispenser, the liquid handler, and how the robots are transferring of the different elements of the assembly procedure. And most of these tools are assisted with our software. We have other workflows such as the cloning, cell culturing, or qPCR. We also do phenotyping, either with a real-time PCR, fermenters, plate readers. And just wanted to give you a little introduction here on the output of what we have and can do. Since in the past 5 years, we have successfully performed 100 large-scale DNA assembly projects. And the EGF, the platform itself, is a really high-throughput assembly platform that can handle thousands of assemblies per week. So our challenge is more to find customers and get them to think big and large-scale rather than actually executing and performing the procedures. We participated in the COVID project during the pandemic, where we have developed the next generation serology test. And more recently, we acquired, with the support and funding of the BBSRC Foundation, a machine that can allow handling of mammalian cells in an automated manner. This is a very big and important field, which is predicted to be growing and estimated to be hundreds of billions of dollars at the moment and growing. This means cell cultures, biologics, antibodies, cell therapies, and so on. This is achieved with the Berkeley Light Beacon system, which is an optofluidic system that can individually manipulate cells into wells or pens in a microfluidic channel or chip setup. And then we can individually distribute, collect, co-culture, stimulate, and analyze the cells. On the right, you can see the actual chip image, which is about half a credit card size. And within that, you can see there is a microfluidic channel, and into the channel there are pens and almost like wells attached, into which we can bring in and out of the cells using light and light capture. And then separately, we can culture the cells in a culture station as well. Just to give you a little oversight or understanding how that works. So it can be used for the cell line development, B-cell antibody discovery, T-cell therapy, and so on. And it can really shorten the timelines from several months to a single week, and also increasing the throughput and the output of the successful clones and hybridomes and so on. So as you can see in the bottom how it works, the light cages capture the cells and move them in or out of the individual pens or kind of wells, and then you can culture them, assay them, treat them, co-culture the cells and so on, and then individually separate and collect the cells into 96 well plates where you can further treat them, proliferate them, or get the genome readouts by sequencing. So as I said, I'm happy to say that most of our pipeline needs have been addressed and supported by our software, and we are in a position now to kind of synthesize them and then put together and build bigger systems now, and also facilitate understanding of the use and the productivity of the software. So I'm going to give a few examples, a few key examples of the main workflow, and then I'm going to talk about the sequencing and also why I think it will be very useful for you as well. You try and use our software and maybe collaborate as well. So the first, one of the first steps is to have a look at the sequence that the customer wants to make and see whether there are any issues regarding DNA synthesis or the cloning, recombination, and perhaps even with the sequencing as well. So for example, Nanopore struggles with monomer repeats, so it's also something good to flag in advance. Hairpins can cause problems with cloning, and so many other difficulties can arise, and obviously you want to flag the restriction enzyme sites as well. We have a software that can, in an automated manner or batch manner, optimize each of these sequences and produce a nice report of what happened. So this happens in the following way. We annotate the GenBank file, the plasmid file, the sequence file, or the BioPython secret code objects with various constraints and objectives, and then let the software work out a solution sequence for each of these sequences that we input. And then it provides a nice report on that, what has been failing before and after, how the sequence looks like, and we have an annotated sequence as well with the mutations introduced and so on. We have also software that can, in a batch manner, add up the fragments for the assembly. We can simulate the assembly. The simulation happens by actually simulating in silico the restriction, the cloning procedure, and finding circular sequences that can assemble together using the overhangs. And we have provided a web interface for most of these software, which can be found at Kuba, the genomefoundry.org. We have about 20-something apps that provide non-bioinformatician, user-friendly interface for many of these apps addressing different aspects of the assembly procedure. You can find our software on GitHub, most of the packages, and we have another website as well that can help you finding optimal strategies for assembling large DNA construct. That means that you can draw up your own supply network for the DNA synthesis, the various tools that you can perform, such as Gibson cloning, Golden Gate cloning, PCR, and so on. And the software will find you the most optimal way to put together your genome, some chromosome or something from the various sequences. This has been mentioned before. Plato is a module that is now part of BioPython, so I'm just going to mention here that we use that to produce the whole plan that can be actually used on the platform, and also a nice report on that, what's going to happen once the actual platform is performed, the assembly. So this is just an invitation and maybe a pitch or an explanation why I think you will be profiting very nicely from using the software. So it would be very useful for us as well to have collaborators or just users to help improving the software and understand where it has shortcomings and where it can be improved and so on. And the main advantage of the software is very useful. It's field proven. Each of these packages, almost each of them, is arising from a need, an actual need from the lab, and has been validated by our operations, which means several thousands of constructs have been made over the years successfully using our software. So it has been validated not only using the software test suite, but also experimentally by our results. It's mature, which means it has been developed over the years, both by the EGF and the wider research community, and it has been used by the wider research community and companies as well, such as mentioning the Ginkgo or DNA synthesis companies such as the Camino Bio in England. And again, all of the software is performing to high software engineering standard. And this is always seen by one of our board members, Johannes Kakadanya, who is a computer scientist. All the software is open source under the MIT license, so you're free to collaborate, work on it, and work it and incorporate into your own system. So if you think anything is useful or would like to try something, then do contact us at EGF Software or whichever our email address is, and let us know if you're using our software. It would be very helpful for us as well. So as a final step, I'm talking about the assembly validation, which can happen either with fragment analysis or sequencing, which can be Sanger sequencing or monopore sequencing. The fragment analyzer breaks up after digestion. We break up the DNA into small fragments and parts which are run on a gel, and this can be interpreted. Our software can predict from the plasmid files the sizes of the band that should appear, and also the size of the band using the data for each of the individual wells on the 96 well plate. And then it can also tell by green and red and other color coding whether the obtained bands are within the allowed tolerance that can be set by the user. We also have other software packages that can help you selecting primers, many primers for Sanger sequencing. And more recently, we developed this next flow pipeline for sequencing analysis. So once we have the DNA assembly, and before that we have simulated the sequences, we have the plasmid GenBank files for each of the products that we are making. And then we are taking the actual DNA and the plasmids onto the Tecan machine to produce the library preparation, the libraries for the nanopore machine. The nanopore is producing the raw FAST5 file and the FASTQ file, the sequence file. So the original plasmid simulated reference file and then the sequence data, the FASTQ file, together with the sample sheet, is used by our next flow pipeline called CQDA that perform the quality control of the reads, the alignment, the variant calling, and then the consensus second generation. And all this data output is put together by our software, a Python package called ADACARA, and that produces the actual PDF report. So the pipeline itself, the CQDA, is an export pipeline, can be run on any platform. It's a one command run that can take all the input data and then produce both the PDF report, but also the intermediate files such as the consensus sequences and so on. Just a quick example here. You can see the size of the reads on the histogram. It can provide various flags, show you the variant code, the mutation table, show you the annotations of the mutation that have been detected, or it can show you as well the coverage of the DNA of the reference sequences. So you can see whether a misassembly or a misannealing of the part happened, something has been skipped. You can compare the length of the reads with the expected sequence and so on. So as a conclusion, I just want to invite everybody again to try our software, collaborate, and I would like to thank our founders and collaborators and the members of the Foundry. Thank you.
Thank you, Peter, for that presentation. I think we'll take one or two questions from the floor if anyone would like to our virtual presenter. There's a microphone in the center there. You can hop up and ask a question if you wish. If nothing on the floor, I'll ask one of you, Peter. Or maybe I might get two in. First of all, how's the Berkeley lights going? And have you tried adapting it for use with yeast and bacteria? Or do you just keeping away from those because it's for your mammalian work?
Yeah, so we have performed many projects on the Berkeley lights. I'm not involved in the lab work of that, but I don't think it's suitable for bacteria. And I don't think we're planning to apply it to the yeast system as well. So currently we're working with mammalian cells and the various workflows that we are currently getting training on as well.
Thank you. And just one question about biosecurity screening. Just how do you stay on top of updating your references for that? Long answer.
Yes, so there's an organization called the IGSC, which is the International Gene Synthesis Consortium, which is a trade organization of the various DNA synthesis companies, and they have a reference database that share between each other, and this is how we update the references. So it's like a community effort, more or less.
So access to a community resource, great. Thank you. Thank you so much for presenting to us today virtually. And we'll say goodbye to you. Hope you can stay online for the next presentation, but we're going to switch speaker now. Thank you, Peter. Excuse me. Great. Thank you. Tong-Si, hello. Thank you very much for joining us today. Can you hear us okay?
Yes, I hear very well. Can you hear me?
Yes, we can. And your slides are up on the screen. I'm just going to give you a really quick introduction. Our final presenter for this session. Tong-Si joins us from the Shenzhen Institute of Advanced Technology. He's a professor of synthetic biology. Tong-Si has an interest in integration of SynBio with robotic automation and high throughput analysis. And some of you may know that the automation facility being developed by SciAT is going to be very impressive, or is very impressive. And so I'm looking forward to hearing about the work that Tong-Si and the team are doing there. So thank you for joining us virtually today, and we'll try and squeeze 2 questions in at the end. Thank you.
Thank you very much for the kind introduction. So today I'm going to talk about our recent effort in the development of automated DNA assembly for protein engineering. I think the previous speaker has given a fantastic introduction on protein engineering, so I will be brief on the background. So as we know, proteins play central roles in life, and thanks to the advances in DNA synthesis, so we are accumulating these protein sequences at unprecedented speed. But however, we are not quite sure what their functions are. Although we right now have the computational power to predict their structures, but either from the sequence homologies or the experiments, still we know only a very tiny portion of these proteins. So we still need these experimental capabilities to characterize these proteins, and in that way, the biofunction and automation can contribute. So especially for enzyme, the prediction of the structure alone is not sufficient, because enzyme, their functions relies on the conformational dynamics. So one way for the enzyme engineering, previously the speaker also mentioned the direct evolution, where we assume no prior knowledge, but depends on this random mutagenesis and high throughput screening, and this cycle on the one hand to improve the properties, and on the other hand also through reverse engineering to understand those. But because we most of the time just accumulate the best mutations at each round, there are the risk of like be trapped in the local optimum. So that's where right now, with the help of this automation robotics, we could accumulate more data, especially those not so good ones. So we could have a global overview of the sequence function landscapes. And from there, we could use the data science to help us to guide through these landscapes to find these global optimum. So in this talk, I'm going to talk about 2 scenarios we have recently implemented on our foundry. The first one is the single site saturation mutagenesis. So like we looking at the 1 mutations at a time. Then like for typical 300 amino acid proteins, the library size is about like 6,000. I think that's still handleable by the most of these biofoundries. And then we're also looking at this multi-site combinatorial mutagenesis, but there's going to be this exponential explosion on the library size. So that's where we are trying to implement the design, build, test, learn cycles, just to like basically to more efficiently guide through these landscapes. So for the first one, we are using the basically very traditionally the very classical basically PCR coupled with Gibson assembly, so where is the degenerated codons or encoding in the primer. So we include them during the PCR processes, and then use Gibson assembly to basically make these plasmids. And then you can imagine like there are the numbers of plasmids libraries we need to make is equals to the amino acid residues we are targeting. And then after that, we need to assign this mutation impact or these sequence function relations. So one way to do it is make basically you do it on the put format. So that's where we put them together and use some assays to link the mutational impact to a fluorescence readout. So there where we can use FACS to separate them into different bins, and then use this sequencing technology to add barcodes into different bins and put them together, use next-gen sequencing to separate them out. So this can be equally applied to where these phenotypes can be linked to the growth fitness. On the other hand, there are sometimes these phenotypes cannot be directly linked to fluorescence or growth. For example, the chemical production. So that's where we probably need to dereplicate these mutant libraries into clones and then put them into these 96-well plates, and then using this PCR to introduce like DNA barcodes to basically code the physical locations, like basically which well or which place, basically barcode that information into the DNA barcode. So that's where after all this put sequencing, we can trace back to where that mutant is from. So I'm going to talk about two applications where we use this, just mentioned this comprehensive single site saturation mutagenesis to create and screen the libraries. So the first example is a peptide antibiotics. So this so-called ribosomally synthesized and post-translational modified peptides, so they contain these basically rings. That's where these rings endow their bioactivities. And the nice thing about this thing is like this structure of these antibiotics is encoded by a gene. So that's where the bioengineering is pretty straightforward by just introduce the mutations into its encoding genes. So most of these antibiotics are produced by these so-called non-model organisms. So the engineering of these organisms is not that easy. So previously we established these systems so where we can recombinantly produce these peptides in E. coli. So I will not go into details, but essentially we could build a general platform to produce these antibiotics. So for these peptides, we're gonna targeting those residues not involved in the ring formation, because rings are important for their activities. So previously we use a manual like colony-based process to do this kind of screening. So basically we use different like induction medium, and also use the temperature change to induce the autolysis of the cell, release the peptide, and then do a screening. So here you can see it's a agar diffusion assay. Like each different, this white dot is a E. coli colony, and then you can also see the inhibition zones. So from there, we could use sequencing, use mass spectrometer, and also use the size of the rings, the zones as a bioactivity to establish the sequence structure and the function relationships. So you can see previously this manual process have this large, basically huge like error bars because it's a manual process, and a colony have these large variations. So then we just use our one of these work cells in our biofoundry to process, to do this plasmid construction. Basically use the Gibson assembly to use this to create this library, transform them into the production host. And then we have two of these processes to do either the multi-top mass spectrometry and or the bioactivity assays, but in the 96-well formats. So you can see we use NNK library, so you have like 32 different mutants, and then it can nicely fits into one plate of these three residues. And from there, we could have this mutation and activity heat map. So these are different residues, and these are the 20 amino acids. And then this color codes the activities. So we also do this intra or interplates variations, and also the correlation between batches. So still we see like about 15% of these variations, but it's a much improvement over the manual processes. In using this process, we have this basically comprehensive library coverage of these single site, and then we also go ahead to target some of these mutant tolerant residues and do this combinatorial mutagenesis. And again, we create the library, scan the mutants, use sequencing to tell them apart, and then get the sequence activity map again. And from there, we could isolate some of these improved variants. So that's our first example. And then we also have another example similar, but this time we focus on the enzyme. So we focus on these so-called rhamnolipids, which is considered the next generation biosurfactants, but currently it's produced as a mixture. So there's a large purification cost, preventing its wide utilization. So the main changes or the variations in the rhamnolipids molecular structures is this lipid moiety. So the chain length is primarily determined by this enzyme, so-called RHLA, which synthesizes these acyl parts. So our purpose is to use protein engineering to control the product or substrate specificity of RHLA. So we could have basically produce one major molecules. So again, these proteins have 296 residues. So basically, we do not change the start codon, but for the other things, we just use this PCR assembly and transformation to create this pooled library. And then we pick basically about 200 colonies per each residue to have a basically 95% probability for full coverage. And then we use PCR to do this pooled NGS. And then from there, we could identify those unique mutants. And then we use this macroplate-based mass profiling. We culture them in 24 well plates because we found 96 well have a large variation. So that's where the 24 give us satisfactory results. And then we use the liquid extraction protocol and then spot the analytes onto a multi-target. So there we could achieve about 10 seconds per sample to finish all the tests in several days versus like the traditional LC-MS method. That's where we're gonna take several months. So using this technology, basically we could have this characterization of every possible single mutants of this enzyme. So these figures, I'm sorry, apologize for this business, but you could see this, again, this x-axis is different mutations and the y-axis is different residues. Basically, we could obtain like three mass spectra per each mutant. And then we have these computational algorithms to visualize these results. So where the circle size correspond to the total production, and the circle color represents the product substrates, the product specificity. So that's where we use this biofoundry to have this global kind of systematic profiling of these enzyme specificity. And then once we have that, we go ahead to do this multi-site combinatorial mutagenesis, because as we know, most of the time, the protein engineering requires introduction of multiple mutations to have the desired phenotypes. It's traditionally achieved by iterative error-prone PCR or multi-site mutagenesis. So for the latter, the library size is gonna grow very quickly. If just 4 sites, we're gonna looking at something like 160 K. So even for MALDI, that's gonna take a large time. So that's where we need to introduce this data-driven and or DBTL cycles to more efficiently create and screen the library. So in this case, we could, from the previous results, we have isolate a few of these mutants, and then we know these residues are important for the specificity. So we're gonna target these 4 sites. So our objectives just try to screen a manageable portion of the total space, and then we could achieve a rapid iterative of these machine learning design and experiments. So as you still can see, the machine learning designs just take a very tiny portion of the time, but the experiments, because E. coli need time to grow, so at least in 3 days, but we can, in the 3 days, can sample about hundreds of thousands. So that's where we do this optimization. But in this case, it's different from previously, because previously it's more like random mutagenesis, but this time you want to make these precise mutations that the algorithms ask us to make. So that's where we need a way to create this any of these 100,000 mutants. So that's where we use a previous robotic workflow for Golden Gate assembly. So we basically first we create these single mutations on different parts. So in total, we create 80 plasmids, and then for this combinatorial mutagenesis, it essentially becomes a linear combination of these different plasmids. So that's where all these 100,000 mutants can be created from these 80 parts. So that's where that's how we can make like hundreds of mutants basically quickly. And then we did a little bit optimization algorithms. We found like the 384 formats is a trade-off between the economics and speed. So that's where we're gonna go forward. So that's the total workflows. So basically from the 80 different substrates, we use the algorithm to generate the pick list, and then we're gonna make 384 different mutants that the algorithms told us to do. And then we do the transformation, get colonies, and then do the induction, and then add the organic phase to extract the products and then use module. So for the cloning and production, it's gonna take 1 day for the ecology to grow, and the analysis typically take a couple of hours. So in this way, we achieve about 7-fold improvement relative to the wild type in about 4 rounds. So we testing about 1,050 mutants to achieve a very quick improvement in specificity. So right now we are trying to look at more sites, larger space, and more smarter libraries by looking at, this time we're looking at 11 sites and 30 mutants and trying to look at the combinations. So again, we're gonna separate these different combinations into different parts, probably 4 to 5. So that's where we can generate these 1,000 combinations from about 100 parts. And also we need the algorithms to do this higher order prediction from this lower order data. So essentially, we can look just comprehensively create these 1 to 3 mutation combinations, and from there, the algorithm could reliably to predict any of these higher order fitness of these mutants. So with that, I would like to acknowledge the group and the collaborators and the funding agency. And that's all I have. Thanks very much for your attention.
Thanks very much, Tong. That was a great presentation from you. Do we have some questions from the audience? Yes. Make your way up to the microphone, sir.
Hey, Rasmus Jensen from LanzaTech. First off, great presentation, very impressive work. In order to speed up your test cycle, have you considered using cell-free protein synthesis?
That's a very good question. Yes, right now we are in the process of developing that. And also we are also incorporate this liposome in the cell-free systems. So we could looking at some membrane proteins as well. So yes, we are, that's gonna speed up the process even more.
Another question from the audience? Any brave souls? I actually don't have any questions, but I took lots of images of your slides and I'm going to review your presentation. Everything's being recorded. So yeah, I'm going to be taking some of these wonderful ideas back to my team. I'd like to say thank you very much to all the presenters today, and thank you for your patience as well, waiting online for us, for anyone who is presenting and also anyone who is in the audience online today. So I'm not quite sure what the break schedule is going to be now because we have still run over time, but I think there will be time for a coffee.
Yeah, so,
and
so
yep.
same as gig as this morning, so maybe what I asked you to use, we gotta catch up a little bit, is maybe you grab your coffee and cookies and then we head back into the discussion in the same kind of run of discussion that we had this morning. And
Yep.
that would followed by presentations from LanzaTech. I see Jake is here from Ginkgo, and Mike is here also from, so we have a few speakers lined up from the industry after the workshop and coffee and cookies. All right.
Great. And thanks to anyone who's online. We're going for a break. We'll be back. Hopefully you're still there. Okay.